we're having technical difficulties. Okay, so fungi is a Latin word. So if you say fungi, 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 as long as people understand you, you can be confident you're saying it right. And I will say it a couple different ways. So tonight I will take you on a tour of fungi, who they are, where to find them, and what are they doing there. Then we'll move to the discussion of conservation of all organisms and zoom in on fungi. What's threatening them and what can we do about it? Let's dive in. So first, what is a fungus? Many people equate fungi to bacteria as microorganisms, but fungi are not closely related to bacteria at all. Looking at this simplified tree of life, you can see that fungi, right here, are a kingdom of life more closely related to animals than plants or bacteria. Fungi are a kingdom of eukaryotic organisms, just like Plantae and Animalia are kingdoms which hold hundreds of thousands of species in each. To get a little technical, fungi are united by their cellular components, what makes up the body of the fungus, which differ from plants and animals. Fungal cell walls are made of chitin, while plant cell walls are made of cellulose, and animals do not have cell walls. And fungal cell membranes use ergosterol for cell structure, whereas animal cell membranes use cholesterol. So there are also similarities between fungi and both animals and plants. For instance, like animals, fungi are heterotrophic, meaning they cannot produce their own food and they cannot do photosynthesis like plants. So they must get their nutrients from external sources. The growth habit of fungi is sedentary, similar to plants. They cannot walk around, and once a mushroom pops up, it stays put. In order to move around, fungi use reproduction and dispersal. Fungi reproduce by spores, which are microscopic seed-like particles, and spores are produced in mushrooms. On a typical mushroom with a cap and a stalk like this russula, the spores are produced on the underside of the cap, generally, though as you'll see today, there are many variable forms of fungi. Spores develop on the underside of a mushroom cap and use very specific physics to be released from the mushroom properly. As you can see here, each gill has many microscopic spores that fall in between the gills to be released. After the spore is released, they are dispersed like seeds, mostly on the wind through the air and sometimes with the assistance of animals. But what is really important to know about fungi, what I want you to take away from tonight, is that the mushroom, or the reproductive structure where the spores are produced, is just a small piece of their body, like an apple on a tree. Fungal bodies live underground or hidden within substrates, such as a log or even a frog. Most fungi that we can see in the world, the mushroom-forming fungi, are called filamentous fungi. They grow by extensive branching networks, a web-like growth form that you can see here called mycelium, which is made of hyphae that are more thin than hairs. This fine hair-like branching network helps the fungus maximize its nutrient acquisition to fully exploit the substrate that it's growing in. So when you see a mushroom pop up on a log or in a lawn, you can be sure that the body of the fungus is hidden, that the body of the fungus is much larger than what you see above ground, and that it is eating that log from the inside out. So fungi come in a huge variety of shapes and sizes, only some of which look like typical mushrooms. A minority of species of fungi are visible to the naked eye. As you can see here, the top two rows of fungi are visible to the naked eye, showing lichens like this orange lichen and this green lichen, showing typical shaped mushrooms with a cap and a stalk like this Mario mushroom, and some amorphous fungi, which you may not feel comfortable calling mushrooms. On the bottom row, you can see pictures taken under the microscope showing spores. And here I'm showing that spore shapes are very diverse, just like fungal species. And fungal plant root symbionts, where this is the root and the fungus is here. This is the root in all of the dark parts of fungus. So these photos demonstrate that fungi are both microscopic and macroscopic, which adds to the complexity of understanding these organisms. Even more impressive to me than their variable forms is the number of species of fungi on Earth. There are estimated to be at least 5 to 10 million species of fungi, And recall, animals and plants have only hundreds of thousands. Though for fungi, only approximately 100,000 species have been formally described by scientists so far. Here on the bottom, you can see 4 examples of the diverse species of fungi: a turkey tail, an elephant saddle, earth star, and dead man's fingers. So I've labeled them with their Latin names, but hopefully I'll be able to say some common names for you throughout this talk too. Fungi are found worldwide and in every ecosystem, from the tropical rainforest to the oak savanna here in Wisconsin, to the ice sheets of Antarctica, to the deep thermal vents in the ocean. Fungi are there. But what are they doing there? As fungi mind their business, living, hunting for nutrients, and trying to survive, fungi serve many ecologies or functions in ecosystems. The three most well-known and best understood ecologies are the decomposers, the mutualists, and the pathogens. For instance, this Helvella, or elven saddle, is a decomposer, whereas the Amanita, the Mario mushroom, is a mutualist, and this orange lobster mushroom is actually a hypomyces fungus acting parasitic on a Russula mushroom. So first, the decomposers, also called saprotrophs because they eat dead organisms, obtain their nutrients from dead organisms. Decomposer fungi are excellent nutrient cyclers, using enzymes to break down large molecules into smaller ones to obtain the nutrients found within. Fungi make up the dominant decomposers on Earth, alongside worms, insects, and bacteria. Here I'm showing Leucoagaricus mushrooms, no common name, found on UW campus eating dead grass, and Obritillaria mushrooms, the wood eaters, found eating fallen wood. Each of these fungi surround the food source with their web-like mycelium, then exude the enzymes outside of their bodies to break the dead food into smaller bits. Then they uptake the small molecules directly into their cell. Next, we have the mutualist fungi. These fungi live in symbiosis with other living organisms. The fungus obtains nutrients from the soil and obtains sugars by trading with their plant partner at the roots. So this Amanita Mario mushroom is called an ectomycorrhizal mutualist, myco being fungus, rhizal being root. So at the root interface, shown in this small picture down here, the fungus trades nutrients with the plant, nitrogen and phosphorus from the soil in exchange for sugars from the photosynthesis from the plant. Lichens are also mutualistic fungi, which live in relation with either algae or cyanobacteria. In a lichen, the fungus provides a structural home and trace nutrients from the substrate that it's growing on, usually on rocks or wood, and the photosynthetic partner provides sugars from photosynthesis. And then there are the pathogens, the least loved group of fungi. Fungi are, unfortunately, the major plant pathogens on Earth. For example, this rice blast disease is caused by the fungus Magnaporthe oryzii, and it is worldwide one of the most devastating cereal crop diseases, resulting in losses of 10 to 30% of the global yield of rice. And especially on animals, fungal pathogens are very difficult to eradicate because fungal cells are more closely related to animals and plants, as we learned. So the antifungal chemicals we need to target the fungal cells, we have to make sure that they don't target the animal cell parts of the fungus, the animal-like cell parts. For instance, this white nose syndrome of bats is caused by the fungus Pseudogymnoascus destructans, and it is likely an exotic species, which is killing many species of bats across North America. Before I conclude this section on fungi, I do want to address the one question I always get. When I tell people I study fungi, I never fail to hear, "Can I eat that?" Because we're usually out hunting for mushrooms together. So my answer is, yes, maybe, but please don't eat that. Fungi are experts at mimicking each other. So there are as many dangerous lookalike species as there are edibles. For instance, this edible chanterelle on the left resembles the poisonous jack-o'-lantern mushroom in color and in shape. We see this with snakes too, where the non-venomous king snake closely resembles the dangerous coral snake. So if you see a fungus that you think may be edible, but you're not sure, you need to consult an expert who can give you a positive identification, or better yet, just treat it like a coral snake and give it a wide berth. So I want to take this moment before we switch gears to tell you a little story about my early mycology career, which Caitlin alluded to. So as I've told you, fungi can be found everywhere that there are nutrients. So the first project I worked on when I was a research technician at Stanford just after I completed my bachelor's was a project based on the finding by microscopy that this particular community of fungi had a very predictable turnover sequence of succession. So to demonstrate, species A pops up first to be replaced by species B and C, and then species D and E round out the community in a short matter of days. But with the recent advent of DNA sequencing and even more thorough genome sequencing, my professor proposed to isolate and sequence the genomes of these fungi to test whether what we can see as predictable is also representative of what grows beneath the surface. Because remember, most of the body is hidden in the growth substrate. The catch was this pristine community of fungi, which is so predictable and interesting, are dung fungi. We call them coprophilous fungi or dung-loving fungi. They grow on poop. So there I was trying to impress my boss and my peers, going out to the field to collect dung for my big first experiment. The Thule elk, where the dung comes from, live on a national protected seashore, which is just a gorgeous place to work. And the elk themselves are a protected species. So I would follow behind the herd at a good distance so I could collect enough freshly dropped dung without scaring or harming the herd. Then I would bring the dung piles, carefully preserved, back to the lab to grow them in mini growth chambers, which I dutifully watered daily. Science is glamorous, after all. The cool part was isolating the fungi, growing them on petri dishes, and watching their mushrooms pop up on the dung and sometimes even on the petri dishes. We found a large overlap of species that could be identified with DNA within the dung and the species that popped up with mushrooms. And I learned heaps of skills and lessons. So the project was a huge success. So now you know, not only do fungi live in all ecosystems, large and small, but you can also get paid to study them in their most diverse habitats. So that was a very brief overview of what I mean when I say fungi and how they differ from other organisms you may be more familiar with. I hope this has given you a glimpse of the fantastic nature of fungi, and I'm going to come back to them later after introducing conservation science. So what is conservation biology or conservation science? Conservation biology is an interdisciplinary science, meaning it pulls from many different scientific disciplines. Conservation biology is an applied science which studies the biodiversity of life on Earth with the goals to protect and preserve natural ecosystems and the species for future generations. So conservation scientists must bridge many disciplines to reach their goals. To break down the goals further, we can describe them as: first, we must document what biodiversity lives on Earth. Then we must investigate how human activities impact species and ecosystems. And third, we must develop and test practical approaches to prevent extinction, to maintain genetic diversity of populations, and to protect and restore ecosystems. You can see here that humans are central to conservation, whether it's when we're negatively impacting species or when we intervene to prevent future impacts. This picture is an example of Kirtland's warbler, a songbird which was endangered starting in 1967, but since then has recovered thanks to conservation efforts. So to be clear, in this description, biodiversity is defined broadly to include species diversity, including all life forms on Earth, genetic diversity, the genetic variation within species and populations, ecosystem diversity, the diversity of habitat types, and functional diversity, the roles that organisms do. We've discussed decomposers and mutualists, but there are also pollinators, primary producers, and so many more roles that organisms can play in ecosystems. These photos show 4 different species of fungi, of chanterelles and black trumpets, that look different but do resemble each other. So the idea that humans should preserve nature has existed for a very long time. Conservation biology formally began as a scientific discipline based on 3 realizations that are tied closely to those 3 goals. The first realization was that human activity had already damaged the environment in the late 1800s, in the heart of the Industrial Revolution. Next, that humans have a civic duty to maintain the environment for future generations. And third, we should maintain the environment for future generations by using scientific, empirically based methods. Again, note the human focus here, but also the focus on future generations, a major priority of conservation. So for a little history, the first laws enacted were to protect land, not biodiversity itself, in the 19th century. And this first law was the Seabirds Preservation Act of 1869. The discipline of conservation began by applying biological principles to prevent the extinction of species. Along these lines, the US Forest Service started in 1905 with the agency's mission to sustain the health, diversity, and productivity of the nation's forests and grasslands to meet the needs of present and future generations. Since then, there has been a focus on macroorganisms as opposed to microorganisms in conservation. And protecting individual species has become a major driving force of conservation, often with the use of red lists. So what are red lists? The International Union for the Conservation of Nature created the Red List in 1964 with the goal, as you can see, to provide information and analyses on the status, trends, and threats to species in order to inform and catalyze action for biodiversity conservation. This Red List holds thousands of species descriptions across kingdoms and whether they are endangered or not. The IUCN partners with many organizations like those listed here at the bottom to acquire data on species and populations. And if you visit the Red List website, you'll find around 80,000 listings for animals, more than 50,000 listings for plant species, and less than 600 species of fungi listed. Fungal species represent only about 1% of the total number of plant species on the Red List. The field of conservation mycology is very far behind the fields of animal and plant conservation, but we won't let that stop us. In fact, can you think of any fungi when you think of conservation? I bet you could name every or almost every organism on this screen, but I doubt many of us could name a listed species of fungus. It's a hard task for me too. Conservation is famous for its charismatic megafauna, including the animals shown here, but plants are lesser known and fungal conservation is rarely even heard of. So why do we need to conserve biodiversity? What are those threats that humans began to notice almost 150 years ago? Well, conservation scientists have studied the major threats to species and ecosystems for decades now, and we know the problems we face. First and largest threat are habitat loss and degradation, when humans change ecosystems for our desires. The second biggest threat is overexploitation. This includes fishing and hunting, logging, and the pet trade. This is also called overharvesting or overhunting. And the remaining top threats to biodiversity are climate change, pollution, and invasive species. Pollution is simple to describe how nitrogen pollution, agricultural fertilizer runoff, and general dumping of waste could affect species. But only research in recent decades is showing how climate change and invasive species threaten native species as well. To return to these examples, we know that pandas are among the world's most threatened animals due to habitat fragmentation and loss. The Venus flytrap plants are being overharvested from their limited native habitat in the Carolinas for the houseplant trade, and the sandhill cranes are only just now returning to solid numbers after hunting almost drove them to extinction. Conservation is a very broad and dynamic subject, which I have barely scratched the surface of tonight. But I want to turn your attention now to build off of our introductions to fungi and to conservation toward conservation mycology. So let's bring in the mushrooms. I want to discuss what are the threats to fungi, what are the challenges to fungal conservation, and what strategies are being used to conserve fungal species. So fungal conservation or conservation mycology is a younger field even than conservation biology. Because the culture and opinion of fungi is different in Europe than here, it's more welcoming there, more mycophobic here. European fungal conservation efforts have progressed much farther than North American efforts. In Europe, there have been efforts by groups like this ECCF, the European Council for the Conservation of Fungi, to determine the status of fungal conservation and what needs to be done. The ECCF has data on conservation of fungi, which helps inform the IUCN, the Red List we talked about earlier. From their website, the ECCF aims to promote the attention for the conservation of fungi to all governmental bodies and non-governmental organizations, as well as to politicians and mycologists. Another large group, the International IPBES, produces sustainable use assessments every few years on the current global state of ecosystem services. The most recent version of this sustainable use assessment, produced last year, was the very first sustainable use assessment ever to consider fungi and their sustainable uses globally. Returning to the red lists, recall that I said animals and plants each hold tens of thousands of species on their red list? Well, when I checked the numbers this morning for the most up-to-date numbers, fungi have a whopping 550 species described and listed on the IUCN red list in 2022, only 476 if you don't include lichens. But before 2020, there were less than 300 species of fungi listed. So the ECCF 2020 Red List Initiative has made great progress in recent years. Most fungi on the red list are mushroom-forming fungi or lichens. Here are two species listed without common names, sorry. The Boletus aurantiosplendens from the Appalachians and Neoalbatrellus subceruloporus from the Pacific Northwest. So what does this look like when you visit the IUCN? When you go to the Red List website and search for fungi, and you make sure that the taxonomy is showing kingdom fungi, this is the first page you'll see. It shows mushroom-forming fungi and a quick overview of if the population is stable or decreasing or unknown, as well as the Red List category, whether the species is endangered, critically endangered, or data deficient. This is a fun, interactive website. If you click on this stats tab right here, we can visualize the data of this website in some interesting, informative ways. For instance, here's the list of fungi organized by their category or level of threat to the fungal species. We can see most species fall into categories of vulnerable, endangered, or least concern, which is good. You can see some species fall under this data deficient category. So we know that even some species listed here do not have enough information on their populations to list them under a more specific category threat level. To date, no fungi are listed as extinct, though that may change with further data. One other cool way that the IUCN shares data is listing the major threats to species. This slide shows the top threats to fungi listed on the red list. And the threats to fungi mirror the threats to all biodiversity. Habitat loss and degradation, climate change, and overexploitation are the top three for fungi. On the website, you can hover your mouse over each bar to see more detail. For instance, these first few bars say housing in urban areas, habitat shifting and alteration, climate change and severe weather, and increase in fire frequency or intensity. We can even see overexploitation represented by unintentional effects of small and large-scale harvests of mushrooms. The challenges to fungal conservation are more nuanced than the challenges to animal conservation, and that's what I'd like to discuss next. So the challenges to fungal conservation. First, fungi are not charismatic megafauna like panda bears and birds. Fungi are largely invisible organisms, only popping up above the surface to reproduce once in a while, not even every year always. Another hurdle to fungal conservation is that people often think of pathogens when they think of fungi, ignoring all the other important ecosystem functions which fungi provide, which we've discussed. To me, this is a misunderstanding and a lack of general knowledge about fungi. Personally, I was not taught a lot about fungi until I was in the university setting. Finally, if we are relying on red lists to define which species are threatened, we need to know not only their population data, but more fundamentally, we need to define what a species is. In biology, there are a few species concepts to define what a species is. The biological species concept uses sexual mating compatibility to define species. It asks, can they make babies? The morphological species concept uses physical appearance to define species. Do they look alike? And the phylogenetic species concept uses evolutionary relatedness to define species. Are they related by DNA? The definition of what a species is may be different across biologists and across disciplines. So if we rely on red lists to protect species, fungi may be left behind. Unfortunately, we are still at such a baseline level of collecting data about what fungal species and populations grow where that we can barely scratch the surface to determine if they are at risk. Protecting individual species of fungi in a parallel manner to animals and plants is fraught with errors until we increase this baseline knowledge. Here is a chart on the research needs and perspectives for fungal conservation of wood-inhabiting fungi, though I find it to be applicable for most fungi. First, we need to know the taxonomy, or the relatedness between species and their close relatives. We need to know the population status. Is the population connected or disjointed? Is it genetically heterogeneous or inbred? We also need to know the basic ecology of the species. What habitat do they grow in? What is their ecosystem function and how do they obtain nutrients? How does this fungus interact with other organisms in the community? We need to be able to answer these questions to conserve fungal species. There you go. Most fungi cannot be cultivated in the lab in petri dishes. So we often can't use fungal zoos to preserve rare or important species like with animals. Although, how cool would a fungal zoo be? Microbiologist James Staley said, microorganisms are threatened when their ecological niche is threatened. The most satisfactory manner in which to preserve the organism is through the protection of the environment and thereby the natural community itself. Data shows that we will have much more success protecting fungi when we protect the habitats in which they live. Fungi need continuous and connected habitat with plentiful nutrients and resources. Habitat loss is such a threat to fungi because removing old or dead trees is equivalent to removing rare fungal species. If you've been foraging for fungi in the woods, you'll notice that most mushrooms do not pop up along the trail where there is foot traffic. Most fungi are sensitive to changes in their habitat. Even soil compaction made by boots can affect them. So protecting entire ecosystems that hold communities of diverse fungal species is key to conservation mycology. Thankfully, in recent years, conservation mycology has been growing. In 2018, the term conservation mycology was coined for the first time. An increasing number of research papers are being published about fungal conservation annually, like this one, protecting rare, little-known, old-growth forest-associated fungi in the Pacific Northwest USA, a case study in fungal conservation from 2008. I briefly mentioned the IPBES assessing the sustainable use of fungi for the first time last year, which means that fungi are finally getting the attention they deserve. Also, the ECCF's 2020 effort added 200 species of fungi to the red list. So there is hope. One paper that just came out last year highlighted the need for community-based learning, especially from non-academic citizens, to actively participate in conservation mycology. So what could be the most limiting factor to fungal conservation? Mycologists, of course. Mycology is an excellent field of study, if I do say so myself, with tons of unanswered questions ready to explore. But as mycologist David Mittner said, fungal conservation can only occur if mycologists are conserved. We need the experts of the field, those who are enthusiastic about fungi, to promote the preservation of fungal species and their habitats and work towards conservation. You could even say that mycologists are an endangered species, though not if my lab has anything to say about it. Here you can see me and my fellow mycologists in action, including my professor, Anne Pringle. So we have learned the threats to fungi and the challenges to fungal conservation. Now, why should we conserve fungi? Well, if my introduction didn't give you a good enough example, let's look at these reasons. There are many reasons in conservation biology to conserve biodiversity for future generations. We can break them down into a handful of categories. Species have ecological value, scientific values, economic importance, aesthetic values, and intrinsic values. And fungi fit into all of these categories and more. Earlier, I mentioned ecosystem services. These are goods and services that ecosystems provide to humans, such as water filtration and decreasing erosion. Fungi provide numerous ecosystem services everywhere they grow. As we've discussed, fungi provide the majority of decomposition on Earth to mobilize nutrients bound within dead organisms. Fungal symbiotic mutualists help the growth of plants or other symbionts. By nature of their mycelial bodies, those branching filamentous bodies, fungi promote soil stability and decrease erosion, and fungi contribute greatly to Earth's carbon and nitrogen biogeochemical cycles. Next, we have the untapped scientific value of what fungi can provide. So far, we already use fungi to produce common foods and drinks like bread and cheese, soy sauce kombucha, and the crowd favorites, wine and beer. New uses for fungi, though, are being explored from innovative companies which use mycelium to replace packing material made from styrofoam and even produce bricks for houses and furniture. Would you sit on a fungal chair or use a fungal table? Other products being made out of fungi include surfboards, lampshades, leather, clothing, and packing material. Houses could be next. I found an article from last year stating that as a building material, fungus is cheap, ubiquitous, and a renewable resource. By dry weight, walls made of fungus are stronger than concrete and have better insulation value than polyurethane foam. Personally, I think if we can use fungi to replace plastic and other materials made of fossil fuels, the world will be a better place. And this is not to mention the scientific value of fungal byproducts called secondary metabolites, which are the organic compounds produced by fungi, such as antibiotics. We can all appreciate the history of penicillin made by the fungus Penicillium, and we can only imagine the discovery of future products made by fungi. And some of this scientific discovery is within reach of the imagination because it is being done currently here at UW in Dr. Nancy Keller's lab. There are many economic values for fungi, including their ecosystem services, but I want to focus here on the culinary use of fungi. As we discussed, most fungi cannot be cultivated easily like plant agriculture, so the wild harvest of mushrooms to be sold to the grocery stores and restaurants is a booming business. For instance, 1 ounce of truffles can be sold for over $170. Morels go for about $40 a pound, and porcini, dried porcini, can be sold for $30 a pound. And there are so many more edible species, I can't name them all. The social and cultural value of mushrooms historically has been strong, including the pastime of mushroom foraging with family and friends. Ancient fungal art can be found across cultures. For example, these Mayan mushroom stones from Guatemala are between 400 and 2,000 years old. Additionally, one glance at Etsy with a search for mushroom will show you endless mushroom art, mushroom jewelry, and mushroom knickknacks. On the left here are bronze cast mushrooms from the artist Emily Arthur's collaboration with my lab here at UW. And finally, we can agree that all organisms have the right to live and persist. It is morally right for us to conserve all organisms. Here I'm showing some examples of how fungi can obviously get along with each other and with others. Fungi are beautiful, fungi are unique, and even, dare I say, fungi are fantastic. So to finish, we have learned why it is important to include fungi in conservation biology. But what can we, scientists and citizens, do for conservation mycology? Well, first and foremost, please spread the word about how amazing and important fungi are. Tell politicians, teachers, your family, everyone about fungal conservation to promote the protection of land, the habitats which fungi live in, and the communities of fungi. You can join a group like the Madison Mycological Society here in town, the National Mycological Society of America, or the International Mycological Association to learn more about fungi. You can take action yourself by finding fungi in their natural environments and contributing to the online inventories like iNaturalist and Mushroom Observer. The Madison Mycological Society has an ongoing community science project at the Arboretum and in the greater Dane County area with annual workshops, which you can join to identify your local fungi and potentially find new species. You can attend seminars about fungi. There are lots online these days, and everyone here tonight gets a gold star. If you have access to a university campus, you can take the opportunity to dive deeper into fungal biology and learn more. You can attend biology seminars, microbiology seminars, even volunteer with a lab. UW is an excellent place for fungal biology, and you can learn more at fungi.wisc.edu. So with that, I thank you so much for your time and attention tonight. I hope you have found at least one thing to love about conserving fungi. Shout out to my whole family for being here, and I am happy to take any questions. Thank you.
Thank you so much, Nora. That was awesome. I can't stop thinking of a tiny zoo of fungi. I love that idea so much. I hope someone does that someday. And yeah, I'll open it up and see. Looks like we have a couple questions in the chat. Paul, did you want to ask your question out loud as long as we're all here together?
Oh, sure. So Nora mentioned those fungi, those wonderful mushrooms you can find in the store, but I was under the impression that the white mushrooms or the crimini mushrooms grow on horse poop. Is that true?
You know what? I don't exactly know the substrate of crimini mushrooms. I do know that there's a mushroom farm here in Wisconsin that grows them at scale. And getting that much dung might be difficult. I feel like they must use soil, or actually when you inoculate fungi, you're often using like barley or oats or some other grain to grow them at such a large scale.
And then Tim, do you want to ask your question out loud?
Yeah, sure. First of all, I just wanted to say that Ben Russell in the United Kingdom, he does fantastic stone sculptures of mushrooms that in terms of the cultural value, you should check them out. Alabaster, limestone, it's wonderful.
Awesome.
Yeah. So I was wondering, is it possible to do things like reintroduce helpful fungi, like say in my backyard or locally in the forest? Is that something that's possible? Can I spread spores in a good way? I don't know.
Okay, so yes and no. So I'll tell you first, there are researchers who are re-inoculating trees with fungi so that the heart rot, which is really declining with forest deforestation, so the heart rot species of fungi can be re-inoculated into those forests. So that work is definitely being done to reintroduce fungal species into their native habitats that they're no longer in. But I would caution you to do it in your own backyard, because here in Wisconsin, we have a problem with the golden oysters, the yellow oyster mushrooms, where that's an instance where the fungus was released into the wild from those propagules that you can make on your kitchen counter. And now it's becoming a problematic species. They're displacing the native white oysters that grow here, and they grow like crazy. If you go to the woods here, you'll find logs full of the golden oysters. And that's a new project that my professor is trying to work on, is what's going on with these golden oysters? How can we handle this crazy situation that's getting out of control? So I would not recommend reintroducing fungi yourself.
And then, yeah, thanks, Nora. Jennifer, do you want to ask your question out loud?
I was just wondering why it was so hard to grow mushrooms in the lab. Is it just the interconnectedness with the environment they can't replicate in the lab, or why it was?
Yeah, that's a great question. So I didn't get into it. I tried to keep it very simple, but fungi often have symbiotic relationships with endobacteria in their hyphae. So when we grow a mushroom in the lab, we try to isolate it so that it's just that species, because we're trying not to have other fungal species competing. We want to make sure that we are just growing that strain of fungus with no contamination. But that means that any symbionts that are necessary for the growth of that fungus can't be grown with it. We use antibiotics to remove any bacteria. So not only are those symbionts missing, but often just the growth media that we use isn't the right growth media for that fungus. So we'll use potato dextrose agar, we'll use oatmeal agar, and fungi, not all fungi eat those substrates. I see Paul has a hand raised.
So I'm so proud that my niece is going to save the world by replacing plastic with fungus. So, but what, and you had those examples of mushrooms or fungi providing structural support for houses and things like that. But plastic has, it's a high strength material that is fairly lightweight. So mushrooms or fungi in general have those types of properties. Can they be formed into the forms that plastic takes in so many places in our lives?
Yeah, that's a great question. So you can grow a mushroom into any mold that you can make. So all you have to do is fill it with those oats or barley, and the mushroom will fill that space because it surrounds the substrate in order to eat it, right? So the shape is not an issue. The strength is also not really an issue, and the lightweightness. Mycelium is actually really lightweight, and when they make bricks out of them, they kind of flash fry it. You know, they incinerate it so it's not alive anymore. And so it's a really lightweight material. The only problem that I've read about is the I don't know how they described it, but it's like the density issue. So they can only pile up so many bricks before the density gets too much, like the strength can't hold. And so what I read was high-rise mushroom buildings are out of the question, but maybe single-story buildings, it's a possibility.
That's so cool. We also had a question that came in, I think just to me. Someone is wondering about spores, and they ask, does it help if we use perforated bags when we're transporting morels?
Oh yeah, that's a good question. Okay, so yes, if you forage for mushrooms and you want to make sure that those mushrooms are there again and again, one common tradition is to use either a wide basket or to use a mesh bag, so that when you're carrying your fungus out of the woods, out of the prairie, out of wherever you found it, then its spores can fall as you're leaving the forest so that it will repopulate. And now, like I said, the mushroom is only like an apple on the tree. So if you collect 5 mushrooms, there's still that entire body of the fungus underground. So over harvesting of mushrooms is hard to do unless you are collecting thousands of mushrooms, you know, hundreds of mushrooms. So we don't want to over harvest fungi, and allowing their spores to fall is one way that we try to kind of mitigate that.
All right, and Kathleen, did you want to ask your question out loud?
Sure. Nora, great talk. Thank you so much. That was so excellent. Yeah, I'm just curious, what is your favorite fungal species and why?
Oh, well, you can see it right here. So here's a Helvella and here's a Helvella. I don't know if you can still see my screen. So my favorite fungus is called the elven saddle. Its genus is Helvella. And mostly I love them because they're whimsical. Like if you just picture a little elf riding on this mushroom in the forest, like what's better than that? But what's cool about these fungi also is that they are in a different phylum of fungi. So most of the mushroom forming fungi are in the group that we call basidiomycetes. And most mushrooms form from that phylum, but the other large phylum of fungi, ascomycetes, which usually, you know, the yeasts are in ascomycetes, usually they're microscopic organisms. A lot of the dung fungi are ascomycetes, they're very small. But Helvella and morels are some ascomycetes that actually grow to a size that you can collect with your hands and, you know, share with your friends. So Helvella, definitely.
Excellent. Thank you.
Does anyone else have a question that they want to ask out loud? I'll just, in the interim, I'll ask a quick question for you, Nora. I know you mentioned that the golden oyster mushrooms that people sometimes grow at home can be problematic. Are there types of mushrooms that you would recommend people try if they're going to grow mushrooms at home that would be maybe a safer option?
Yeah, that's a great question. Okay, so my second favorite fungus is called Hericium. It's the lion's mane or the bear's head fungus. And they are native to Wisconsin. You can find them on old logs, especially in northern Wisconsin. And they are another one where they sell the kits to grow them on your counter, you know, kind of in the dark corner of your kitchen. And lion's mane fungus, it's, some people use it to replace crab in crab cakes. It's kind of got more of that seafood flavor to it. And they don't look like mushrooms at all. They've got a tooth-like growth form. Sometimes it looks like a waterfall, like a frozen waterfall, which is really pretty. If you're interested in fungi and you don't really like the grocery store mushroom because they're bland and boring, try out lion's mane.
Hey Nora, is that a woody mushroom, lion's mane?
Do you mean like a decomposer mushroom?
Yeah.
Yes, yeah.
Oh, okay.
And when you're growing mushrooms in your house, pretty much all of them will be decomposers, because if they're that mutualist fungus, then they cannot really survive without their plant mutualist. So if we're growing mushrooms, you know, from a kit, then they're going to be decomposers. Yeah.
Oh, that makes sense.
Yeah. All right, Sean, I see your hand is raised.
Yeah, thank you for sharing with us tonight. I just have a question about like harvesting locations. If you consider it, I guess I've heard like fungus, they will like absorb heavy metals and stuff like that. Would you consider it safe to harvest mushrooms in like the city parks? Maybe like, I don't know how far from roadways and stuff like that is that?
Yeah, that's a really good question. You're very right to have caution about, pollutants that may be in fungi, because like you said, they can uptake heavy metals. They are experts at extracting nutrients, and the heavy metal, is a nutrient to them. So one story that I have, the chicken of the woods, Laetiparus fungi, when I lived in California, they grow on eucalyptus trees, whereas on the East Coast, around Wisconsin, they grow on hardwood trees. And so on a hardwood tree, the chicken of the woods is delicious, highly edible, no stomach problems. But my friends who ate chicken of the woods that grew on eucalyptus, because like you said, eucalyptus are a species of plants that hold those heavy metals, those toxins in their bodies. The chicken of the woods on eucalyptus caused some gastrointestinal distress to the eater. So it's really case specific. You need to be knowledgeable about what species it is you're eating. You need to know where you found it. So you're not right next to the dump. I would not suggest eating mushrooms found right outside the dump. And then just be cautious. So if you have a mushroom that you know what species it is, it's edible, just eat a few bites and wait to see how your body reacts to it. And then if your body reacts to it okay, then you'll know it's safe to eat for the future. I try to give extreme caution about eating wild harvested mushrooms because, like I said, there's so many lookalikes that could be bad for the gastrointestinal system. Paul, you've got your hand raised.
Didn't I read something recently about how Portland is decriminalizing psilocybe mushrooms? So is Madison going to catch up?
Madison is right on board. There is a new master's program at UW-Madison in the College of Pharmaceutical Sciences, trying to study the psychoactive effects and the pharmacological effects that we can use from psilocybin mushrooms. Yes, so that's another one where it's like, use extreme caution, but studies are definitely showing that there are benefits of microdosing and benefits when you're in that, safe practitioner setting.
I'm going to jump in for a moment. I might ask a question about morels. Speaking of this last one, a lot of times they go to railroad tracks, and railroad tracks are kind of skeptical. Morels growing along them?
Yeah, is that a question? Sorry.
To be worried about morels along railroad tracks or other areas that might be kind of high exhaust or like roadsides?
Yeah, that's a good question. You're right, because morels often grow in what we call disturbed areas where either a fire has come through and then you'll see a whole flush of morels pop up, or you'll bring in new mulch and then a bunch of morels pop up. And just like you, I found a morel next to a railroad track before, so that's really funny that you mentioned that. Yes, use caution for sure next to railroads. I would say it's a great specimen to collect for, to show all your friends and to take pictures of.
Well, the dead elms, and they're yummy as hell.
Yeah, and in Wisconsin, just go out in May. Keep trying, It
used to be April.
Really? Oh, that's exciting. I've only been here 5 years, so it's always been May for me.
Awesome. And it looks like we got one question that came in through Facebook, so I'll just pop that one in. Someone's wondering, what is a good starter mushroom for people to look for in Madison?
That's a good question. Okay. It depends on the season. Sorry, there's so many fungi. It's really hard to pick one. So, okay, if you're in May, look for morels, right? There's also what's called a pheasant back mushroom that happens at the same time as morels. And it looks like the back of a chicken, so it's kind of cute. Let's see. And then I guess one it's kind of everywhere during the summer months are the oyster mushrooms, either the white oysters or the yellow oysters. And I guess if you just Google oyster mushrooms, you'll see that their gills like continue down the stem. So they do have really distinctive characteristics. But maybe be cautious with that one since it does have lookalikes.
Awesome. Thank you, Nora. Just a couple minutes left. I just want to toss out if anyone else wants to ask a question, now's the time.
I just wanted to jump in and ask if there's any purpose to bioluminescence in fungi. I know that some species bioluminesce, and I was just wondering about that.
Oh, that's a great question. That is an open scientific question, the purpose of bioluminescence, but it's fantastic to find. So the Omphallotus mushroom, the jack-o'-lantern mushroom, is one where if you take it into a dark room or have it out under the stars, it will glow, kind of like a whitish bluish glow, and highly recommend if you can find it. But I have not read the most recent literature, so I'm not sure about the reason behind it. Maybe for insects to find the fungus so that they can spread the spores. It's a good question.
That is a good question. Thanks, Maggie. All right, anyone else? You've maybe time for one more question. So if you've been dying to ask, now's your chance.
Yeah, I guess I have one question.
Okay.
Since it's Audubon, I mean, do birds in Wisconsin have a relationship with mushrooms or fungi that we should know about?
Ooh, that's a good question. So the one bird that I know of that's got a relationship with fungi are woodpeckers. So the woodpeckers that bore holes into wood, they have special communities of fungi that live in their beaks, and then they are responsible for spreading those fungi. So they are dispersers of those fungi, which is really cool. I did try to throw in a lot of bird pictures. I hope y'all noticed that.
I noticed.
Okay, good. Yeah. But yeah, woodpeckers. It's a new discovery, which is really fun.
Oh, that's super cool. I totally didn't know that.
That's great. Thank you.
Thank you, Nora.
Yeah, thank you so much for all your questions. It's been a challenge, but very fun to try to answer them.
All right, well, I think that, yeah, we'll maybe start to wrap up then. I just want to say again, thank you so much, Nora, for being here tonight. It was awesome to learn about fungi and conservation. I had no idea about the woodpeckers. I'm so excited to know that new fact and share it with everyone I meet. That was fabulous. And thanks so much to all of you for joining us this evening. Hope to see some of you at some of our upcoming events. And yeah, thank you all so much for joining us. Take care, and I hope you all have a great night. Thanks for coming.
Thank you so much.